Yeah, thanks for inviting me to ICTS. And so today actually it's mostly going to be an expository talk, and interspersed within it will be stuff that I have been working on. So sequence alignment is a very fundamental problem in computational biology or bioinformatics, you could say. And that kind of drives most of the work that goes on in this field, like directly or indirectly, you could say. So I was told that not everyone here has like a background in genetics or anything. So I will be covering some basic concepts before we dive into what exactly sequence alignment. We need to know what a sequence is and what an alignment is before we do that. So this is the central dogma of life, you could say. Like you have the DNA template, which has is actually the same for all cells, but one expresses RNA. RNA is what kind of drives what kind of tissue you are, and the gene expression is based on RNA, which gets translated into protein through the ribosome. Here you see a picture of a ribosome, and the mRNA is being captured by the ribosome, and all these tRNAs. And this is the translation mechanism. There are codons. I don't think I mentioned that here. So if you think of the DNA just as a string, and you must have heard in popular science books that 98% of the DNA is not really coding for any protein. 2% of the DNA is what's doing most of the work. I mean, technically speaking, that's not quite true. There are many regulatory functions in the other 98% of the DNA. But what is meant by that 2% is this DNA is first transcribed into RNA, and RNA essentially. Let's look at a picture. I think my next slide has a nicer picture. So let's say this is the part of the gene, part of the DNA, which is the gene, and these are the exons. And when I say exon, these are the actual parts which eventually merge to form this RNA. So eventually there is something called a splicing mechanism, which removes these interior parts, and which are called introns. So these exons put together form different types of RNAs. Here you see, you don't see the mRNA here. So whenever we talk about coding, it's about messenger RNAs. So those are the ones that get into the ribosome and translate into proteins. And the code essentially is a triplet, for example, let's say ATG in the DNA. So let me just write it ATG. For example, this triplet corresponds to a certain protein, certain amino acid, which is actually methionine. I mean, the details are not that important, but this is a very special kind of codon, which is called the start codon. Anytime a transcription start in a transcript, the coding starts, it's from the start codon. But it's not necessary that the start codon is from the very beginning. You have untranslated regions called UTRs at the beginning and the end. So you have 5' UTRs, 3'. DNA is kind of oriented. There's a 3' end, and there's a 5' end, as you can see mentioned there. So you have these untranslated regions, which actually have promoters. They have signaling pathways. They have signaling motifs, which tell where to start your translation. Yeah, and as you can see, from what I said, every triplet kind of codes for an amino acid. So the coding part of the RNA is essentially a multiple of 3. And how does the ribosome know when to stop? There are certain codons. There are 3 codons known as stop codons. Although strictly speaking, again, that's not true. More recently, in the 10-15 years, there's a 21st amino acid that's been discovered. In general, usually in older textbooks, you'll find there's 20 amino acids, but there's a 21st, which is called selenocysteine. And one of the stop codons actually codes, given there is some other regulatory element called the cis element, so it can actually code for some amino acids. So this is the basic central dogma. DNA becomes is transcribed to RNA. RNA is translated into protein. And of course, there are some exceptions, you can reverse transcribe RNA to DNA. So the central dogma doesn't capture that, but this is the very classical picture of how things work. And as you can imagine, maybe what you're probably most interested in functionally are proteins, but they're very much, the recipe for the protein is kind of contained in the DNA. So here, these are the 3 things that are determined by sequences, and that's where the sequences come from.
So what are these exons and extrons?
Yeah, so if you think about the genes are actually, it's not like 100% well-defined thing. You can think of it as a region of the DNA. And so these exons are the parts that are like coding, that are the functional parts of the gene. You can think of it that way. They are the functional parts of the gene, and these introns are what are spliced out. Like in the nucleus, first you transcribe the DNA, then before it comes out of the DNA, this whole chunk will be called the pre-mRNA. And the pre-mRNA, there is a splicing mechanism which removes these parts so that these three join together to form the transcript. And so these introns are not very well defined because you can have a gene sitting inside this intron. But yeah, that is possible. Like introns can be huge. Exons are usually 100 to 200 bases long. So you can have another gene. So introns are relative term. Yeah, another thing is because of this phenomenon, you can have something called alternate splicing, meaning you have, like for example, there can be exon skipping event. Like you ignore this, the whole thing is spliced out. So a single gene can actually have multiple transcript expression, multiple isoforms, so to say. So that's why we have about, yeah, just let me finish this. You have about 20,000 genes, but about 80,000, like humans, have 80,000 active transcripts because a single gene can actually code for multiple transcripts. Yes?
Structurally, in terms of base pairs, there is no distinction between
two
regions.
Two regions, meaning, I mean, I have just given you one strand. I haven't really, yeah, a gene is kind of gene actually belongs to a particular strand. Like there's a plus strand and a minus strand of the DNA. So you actually know where the location of the gene is because the promoter regions are in there. It's not like, so if you think of it this way, let's say you have some string A, C, G, T, A, G, something like this. And then you have, of course, the reverse complement right here. You have C, T, T, A, and so on. So when I say a gene, I usually know which strand it's coming from.
But if I take this one strand, then structurally, is there a distinction between what you call exons and
oh, you mean like in terms of DNA structure, like physical structure? No, in physical structure, there is no difference, yeah. So, yeah, so from the central dogma, we see these are the sequences of interest. We have DNA, the alphabet has 4 letters, and you have RNA, and the T, the thymine, becomes a uracil, and the protein, will be flexible. We'll say sometimes 20, sometimes 21, because sometimes, the bacteria actually have, again, there's another stop codon they can use. So, as in biology, all the time you have exceptions. So, yeah, we'll be flexible on the alphabet here. So a gene or transcript becomes active or expressed in a cell by being transcribed RNA. That's what I said. The mRNA is code for proteins with each triplet of bases in an open reading frame being this thing. Because you can, if I had this sequence, let's say A, C, C, T, A, A, G, if I started reading from here, this is a reading frame, triplets like this. But let's say I started reading from here, I would consider triplets like that. But of course, if it doesn't start with ATG, then that would not be a start codon. But the point being, there are 6 reading frames, one on each strand, like 3 of them, and 3 of them on the other strand. So there's 6 reading frames. So that's why I say with each triplet of bases in an open reading frame being translated to a corresponding amino acid. Okay, and this is a picture, and you can see what I talked about, splicing, alternate splicing. So this wild type protein just means the normal protein. So you have removed all the introns, you got the usual protein, then you have exon skipping events where you ignored one exon, then alternative 5' splice site. So 5' splice site should be around here. Sometimes there might be an error and you have an alternative 5' splice site, and same with 3', and intron retention, and all these events. And some of them can be.
RNA transcript is the one which triggers this process.
There are many proteins. I forget the exact mechanism. There's 8 to 10 proteins. So the splicing mechanism itself involves a bunch of regulation and proteins, and that happens within the nucleus. So, did I answer your question?
I'm saying, what is the mechanism by which one knows which ones to skip and like how to skip the exons and.
Yeah, I don't.
Comes and it's copy. No, it's not that, right?
Sorry, can you repeat that? I got the first part of your question. How do I know which of these transcripts to choose?
Like, yeah, how to.
Yeah, I'm not 100% sure, but I mean, I guess the easy answer would be to just say there is some like regulatory thing that like if it's there, it does this. Yeah, I mean, I guess I wish I knew, but I don't have a very like concrete answer for this. Okay, and of course, another interest of looking at sequences is if there are mutations, then that can cause a lot of like different kinds of diseases. So that's another interest. So small-scale mutations, you have insertions, deletions, substitutions. So insertions just mean that like you have the usual sequence, something like this, and you have added some, let's say a G here or something like that, by some mechanism. And deletions just means, again, like it's kind of like an opposite of an insertion. One of them has been removed. Substitution means, let's say the first A has been replaced by some C or something like that. That's a substitution. And you can imagine that insertions, deletions actually affect the reading frame. They actually shift it. After a while, like you have 3, 3, 3, and 3, so on. If I delete something, the frame after a while has completely shifted. So the proteins that you'll be coding have completely changed. I mean, the amino acids that's being coded by this transcript has completely changed. So yeah, I mean, it can be a big cause of diseases. And same with SNPs. Sometimes SNPs can be like harmless, because as you can see, there's all these triplets, there's 4 choices, right? And so there's like 64 choices of these triplets. Like I was saying that ATG, you have 4 choices here, 4. So, but there's 20 amino acids, roughly. So there's a lot of redundancy built in. So sometimes even if you have a substitution, it doesn't change the amino acid. But what it can do is, again, do some sort of, so it's, like theoretically you would think it's exactly the same thing, but that's not how it works. Sometimes what might happen is it might cause some sort of regulatory effect and lead to low expression of that RNA. I forget the name of the disease, but like there is a disease which is purely caused by a SNP, which does not really change the protein, but it changes the amount of expression of that like transcript. Yeah, and of course large scale, so these are very small scale, very localized mutations. You have large scale mutations. You have gene duplication, like chromosomal rearrangement. Loss of heterozygosity just means that like almost every gene, you get a copy from your mom and your dad. Like, so you have 2 copies, that's the hetero part, then like maybe one of them is completely dysfunctional, but the other part is still working, so you're still fine. Like maybe a big part of the chromosome has been deleted from one of your, from the pair of chromosomes. Okay.
What are the causes of large-scale mutations? Small-scale mutations presumably can be caused all the time by various...
By random processes, yeah, by chemicals or...
Large-scale mutations happen at the time of cell division or...
Yeah, I mean, like first of all, when you're dividing, like a lot of homologous recombination is happening. Like that is one, like in the sense, you have all this, like before, like somatic cells are being formed, like they're haploid, right? Like we have pairs, but like our germ cells, they just have 23 chromosomes. So when that is happening, before, it's not like you get half from your dad and like in the germ cell, it's not like it's half from your dad and half from your mom. Before the splitting happens, there is a recombination of these pairs. Like, so I guess the picture would be more useful. So let's say I have this normal cell with, this is a pair, let's say chromosome 1, like chromosome 1A, chromosome 1B. So these are the alleles, let's say 1A and 1B. So when like meiosis happens and the cell divides, it's not like you get a 1A in one of these haploid cells and 1B in one of this. That doesn't happen. Before that happens, before meiosis happens, a homologous recombination of these 2 happens. So that's why you have diversity. Otherwise, like you can probably imagine, like it's almost, after like 3-4 generations, there's no mixing going on there. So yeah, during this recombination process, it's not random. The recombination is not random. There are some regions which actually go together. So that it happens in chunks, but like some of them might just be deleted completely. And so a lot of times also what might happen is duplication events might happen. Like duplication would, for example, I guess duplication would not, yeah, would count as a mutation, yeah. Like for example, what might happen is, like you just reverse transcribe by accident, like something to the gene. So that's how you get something called pseudogenes. Like the same gene has multiple copies. Sometimes you need multiple copies because, like for example, ribosomes are very important. So you need like 300 copies of ribosomal gene, functional copies. But sometimes what might happen is you have transcribed the gene, and then you have, by some error mechanism, you have transcribed it back. Now, but it doesn't have the promoter region, nothing. It just has like some sort of like half part. Like it cannot like transcribe by itself, which is like a pseudogene. So, and that causes problems in alignment and all that because it looks like a gene. So, okay. So what is sequence alignment? So sequence alignment is the problem of comparing, you could say, different DNA or RNA or protein sequences to identify regions of similarity. And pairwise is, as the name suggests, you take two sequences and you want to compare both of them. And multiple sequence alignment to align all of the sequences in a given query set, but like there, this question could be a bit more abstract, like in the following sense. So in the first one, when I said pairwise, you have like a certain like database sequence, you have a query sequence, you are kind of trying to align to that. But in multiple sequence alignment, like you could imagine, you don't really have like some master sequence. You're kind of trying to find like what is common between them, like some sort of like alignment altogether with, yeah. So in some abstract sense, which will be more clear when we talk about like some models for like protein family domains and all that. But yeah, you just want to find like some commonality between all these sequences. Okay, and why care about sequence alignment? Many reasons. So for example, one of the very basic things is genome assembly. Like before you can even do any kind of interesting like genetic studies, at least in a high throughput manner, you need to assemble, you need a reference genome, like let's say human or mouse or Drosophila, whatever organism, you need like a reference genome to which you can compare things. And to build that, like it's not like you take the massive, like whatever, millions or billions of base pairs and try to sequence. What you do is chop them up and sequence those. That's what we're capable of, like thousands, 2,000 or 3,000 bases. And then you have to assemble them. That's a big problem. And there, of course, like you do some sort of like end-to-end kind of alignment, and then you piece them together and form the whole sequence. And that's like a big industry in itself. And study of gene or transcript expression using RNA-Seq data. Yeah, so this is actually a big part which is used in biomedical research. So effectively, what happens is like you have a tissue, and ideally you would like to measure protein levels to figure out like what's going on. Like let's say you have a control thing and a treatment group, and you want to compare like what's different between those two. Like maybe you treat it with a drug or any other kind of experiment. And ideally for functional reasons you might, or mechanistic reasons, you might want to look at proteins. But proteins by their nature, they're very reactive, and like it's very hard to like quickly capture them. So as a proxy for proteins, like recall the central dogma, you have DNA becoming RNA to proteins. So as a proxy for proteins, you can try using RNA, like trying to take a snapshot like of the RNA, and that is something we have been successful at. So that's what RNA sequencing is all about. Like you freeze the tissue, you like prepare like libraries out of like the RNA in there. Like there's a whole mechanism, and eventually what you get, like what you end up with is reads. You don't know where they came from. All you have is like let's say 100 base pair reads, and what you really want to figure out is which transcript did this read come from? Like which part of the genome did this read come from? And then we want to say something like, okay, so looks like all these reads came from this particular transcript, so the count is, let's say, 100. For this transcript, the count is like a 50, and so on. So then you have a spreadsheet, okay, it says that, okay, all these transcripts get these numbers in this condition, for the other condition, a similar number, and you want to see what's different, what has changed. So here, before you can do all that spreadsheet stuff, you have to align. And this is like, you have a massive sequence of 3 billion base pairs, and you have 100 base pair reads. And although that might seem that it's too small to be uniquely aligned, but you can quickly compute that if the genome was random, like what is the probability of like a particular given sequence of 100 things occurring? You'll probably imagine like 1/2 to the 200, right? Like 1/4 to the 100. So, which is like a much lower number than what like you'd see like from the expected value, like randomly sampling from 3 billion things, like contiguous sequences. So now, of course, the DNA is not random, but that's another story. But you can at least convince yourself that there can be some degree of success using RNA sequencing data. Okay, another thing is locating domains in protein sequences. So let's say you have you have done a lot of hard work, like manually, empirically curating, like, okay, these proteins I know really well, these are the domains. Now I have discovered a new protein. So what is the function of this protein? To find out, like, you probably want to align these amino acid sequences and figure out, okay, all these domains occur in this protein, so this is my best guess, kind of, for like what this protein is supposed to do. And of course, you have to validate that, but it gets you started. And establishing phylogeny, and so here you compare like so genes across species, so to see some sort of homology and try to build a tree. Let's say you want to see, okay, this is the kind of parent, and like these all species kind of evolved from that. So you want to see some sort of homology there. And then genome-wide synteny, I don't think this is a very popular field, but there is some interest, and this synteny is effectively about gene co-localization. Meaning, whenever all these recombination events happens, they're like certain genes, they come together. And let's say you compare, let's say, mouse to human. So you can actually see that like a group of genes in mouse, they will be spread across 3 or 4 chromosomes in human, but like they'll be very localized. And people try to study that to see like how evolution happened and so on. Like how all these recombination events have happened across these years. Okay, so now let's get down to pairwise sequence alignment. Okay, there are two notions here. One is global alignment versus local alignment. So in global alignment, we attempt to align entire sequence. We have a database sequence, a query sequence, and we want everything. We don't want any compromises on that. So that is only good for if both sequences are comparable in length and quite similar. So in that case, you can be very stringent about what you want, and of course suitable for closely related sequences. So then you don't expect too many changes to be in there. So you can be very strict about what you want. But what is like the most useful thing, of course, like global alignment techniques were invented first, and then they were adapted to local alignment. But in a more direct fashion, local alignment is what is actually directly used. So it finds local regions with the highest level of similarity. So, like for example, when you're doing multiple sequence alignment, you can imagine, like this is pairwise, but let's just like go ahead and think about that. So you cannot expect that a given gene for a mouse and like for whatever function would be exactly the same as for the human. Maybe so, like the more important parts, like there are some like conserved regions in those genes, and that's because when you think of the protein has also a 3D structure, right? So not all the amino acids are actually equally important. So some of them are wrapped in, like some of them are like, some of them are functional, so to say. So if the mutation happens in a part where like it doesn't really affect the function of the protein, in that case, I mean, it's like it might survive, that mutation might survive, and over the time they will diverge. So if we want to find that, we cannot be very strict. We want like local kind of homology. Like we want, okay, this part is homologous to this gene. Maybe there's a big chunk which is not the same. Then there's another chunk. So local alignment is what we generally are looking for. So here in this picture, like, so global alignment is very strict. Everything kind of has to look the same. But here you see, like this is different, but still this will be called a good alignment. This is the same, this is the same, this is different, but this is something we still want to call as an alignment. And this is a very 1970, as you can see, and in math years, 1970 is not that far back, but in terms of these areas, 1970 is classic. So this is a very classical algorithm. Needleman-Wunsch algorithm, and this is essentially a dynamic programming algorithm. So what you do is you have, let's say, for example, you have these two sequences. GCA-TGCU, GA-TT-ACA, and you want to, so if they are the same sequence, of course, you want to call that a perfect alignment. But here, what you want to do is, anytime there is a match, you want to give it a good score, +1. Anytime there is a mismatch, you give a -1. And indels, let's say you penalize with a -1. And of course, these scores are subject to other like empirical knowledge you have. And like here I mentioned that so a more realistic model might be more penalty for the first indel meaning insertions and deletions, in a series compared to the successive ones, because it is likely that if there is an indel, it's bad, but like maybe when there is a deletion event, it kind of has a span. Like if there is a deletion event, there can be more. Like as a chunk, it's removed. So in that sense, you probably don't want to penalize every single one of those. So you can play around with this, and playing around is important because people try to empirically find what makes sense. Like especially when like you talk about like protein sequence alignment, there you have all these matrices, substitution matrices, where they give scores for every pairs of amino acids. But yeah, for a very like toy model, you can imagine, okay, match is good, +1, and anything else is penalized. And then what you do is, just imagine this is like the empty sequence. So empty sequence, there's the one mismatch, -1, then there's two so here you just imagine that we are progressing like inductively in some sense. So if the sequence were just G and you have the empty sequence, you'll get like a deletion event kind of, so -1. If it's GC and you still have empty, it's -2 and so on. That's why these two are, you can trivially fill them up. So now what you do is to fill this up, you look at these two, G and G, so there's a match, so that's a 1. And there's no way to increase that score. So you just keep it as a 1. Now you want to map GC to G. So what are the possibilities here? So you have, yeah, it's better if I draw this. Yeah, by the way, so one thing to keep in mind is, so you have GC here. So if you did this, what would be the score? Be -1, -1, and -1. So that would be a -3, right? Yeah, because, so essentially we're proceeding inductively, right? So here all I'm doing is taking the sequence GC and taking the sequence G. So we'll actually kind of build up a recursive kind of thing. So if this is GC and here we have G, so this is like an insertion.
So what is the top corner? I didn't understand what the top corner.
Oh, okay, so the top corner is, let's say this is the alignment we built. Like, okay, we treated this as 2 deletion events and 1 insertion event. Like G has been, like this is, like, yeah, I mean a very puzzle solving kind of way, mathematical kind of way. Like essentially what you're saying is, okay, if this were the alignment, what would the score be? So this would be a -1, this is like an insertion here. Like you did 2 deletions here and then you inserted G. But the other option is this, which is +1 and a -1. This is -1 and a -1. So this is the best option you have. That's why you get a 0 there. So anytime you see an entry, let's say I show you the entry here, effectively I'm comparing this and up to this. So for this entry. And if you see a 0 here, that's essentially saying that the best score you can get of aligning these two is 0. Yes?
Does it mean that once you move to a field, you add to the upper sequence what is in the column and to the lower sequence what is in the row?
Yeah.
Like when you move from 0 to -1,
Mm-hmm.
you add in the upper sequence a gap and in the lower sequence G. That's the reason why it's -1. Or...
Yes. Yeah, exactly. So this is the empty sequence, right?
It means if you then would move once further down, then your lower sequence would be gap, GA, and your upper sequence would be gap, gap, gap. if you move to the to this -2 in the first column.
So in the first column, -2 here.
Yes.
So this is GA and this is nothing.
And then you would have 3 times nothing.
3 -2, right?
If you go from the 0 to the -1 to the -2.
Sorry, I didn't get your question.
GA and gap, or just GA and gap, which you're comparing for minus.
So GA and gap, this or like you're saying this? So is your question this? So is your question about this? Like,
Yes. So does it work like this or?
actually, this is kind of like an empty match or something. Yeah. So I'm going to say like no, you don't want this, like because you can always do this, right?
Gap doesn't get a +1. Gap.
Yeah, I mean, yeah, it doesn't get a +1, but like when you talk about gaps, you can put many of them, right? Like, so yeah, you don't want to consider that. So here, like, so how you build this up is effectively you say, let's say I know how to align these two things. Let's say GACTG and GATTA. So let's say we go up to here. That's the best score we have. So when I'm aligning this GCA-TGC to GATTA, so you have 3 possibilities. Like you take this best match to Yeah. Okay, yeah, sure. So, so you get a recursive relation. You can build some sort of recursive relation, and you have 3 options. Like you can build from to get the best score here. You can either do a deletion. So deletion events will be this diagonal, as I mentioned here. No, sorry. This vertical will be a deletion event, and this diagonal will be a match or mismatch. And you have 3 options, and depending on what you have, you add a +1 or -1. You have 3 scores, and you take the max of those scores. So that's what you put here. And once you finish this up, you do a traceback algorithm, like from the right-hand corner to the left hand. Okay. And the Smith-Waterman algorithm is effectively it's the same, but just that like anytime a negative, so here you have a bunch of negative scores. Anytime a negative score is encountered, replace it with 0. Thus, local signal is captured by not penalizing mismatches or indels. Okay, so that's the. Okay, so this is something more recent. This is actually 2016. This is not my work, but this is Leo Pachter's work. So pseudo-alignment for quantification. And before I talk about like what is like pseudo-alignment, I need to talk about this particular problem of quantification. So here it is about like what I was talking about was RNA sequencing, right? There you have all this control and treatment group, and you're freezing the tissue at a particular time, and you're measuring the RNA levels. That's the goal. And the way you do it is split the RNA up into tiny pieces, and maybe you get like fragments which are 300, 500 bases long, and there's paired end sequencing or single end, like you get like 100 or 150 bases from each end. So eventually what you get is a file which has all these reads, and has no information about transcripts or anything. All it has is 100 base reads. So now what you want to do is map them back to the genome, so, or rather I should say transcriptome, and you want to know where these reads came from. And that's how you'd be able to quantify what was the expression level of those transcripts. Does that make sense? Okay. So, yeah, so there are aligners which actually like do the complete genome level, and they have all these indexes which relate all these exon maps. Like for each gene, you have the annotation, you know where they occur, and they do the alignment. but they take a long time. Like in the sense, like to give you an estimate, they require like 50 GB of RAM. There's a tool, a aligner called STAR. It has about, like it requires a lot of resources, and it takes about, actually it takes about half an hour to run, but like 50 GB of RAM is already like very resource intensive because it has to build a massive index. So, but the point is we have already gained a lot of information over these years, so we can actually kind of use that. Like we don't have to assemble anything, we don't have to annotate anything, that's already available to us. All we want to do is quantify. So for that reason, we want to, like maybe there's more information which you can use to like speed up our like quantification process. And the way that Callisto does it, so it's a transcript level quantification tool, so you can measure transcript expression levels. And so what it does is for a given transcript, you know the sequence from the reference genome, so whatever, like 1,000 bases or so on. And then it draws what is called a transcriptome de Bruijn graph, meaning it takes k-mers, usually like k equals 30 or so. And if you shift this like a window, like this 30, shift this as a window, and you have, what are the possibilities of these k-mers? That's like 4 to the 30, right? So now you have a graph, and each node of this graph, so node 1, and so on, there's going to be many nodes, and each of them is a k-mer. And you draw a line from this to this, if the last 29 of these are the first 29 of this. So essentially it's like, by just sliding, you can reach to N2. So anytime you encounter a transcript, it kind of gives you a path in this de Bruijn graph. And what Callisto does is, now it looks at the reads. Now it's, okay, for the transcript you have this, from the like annotation it builds an index. And then once you have that index of all these paths, it takes, like from the read, like a 100 base read, it starts taking all those 30 mers, so to say. So tiny 100 base read. So you have, let's say, like 70 odd like k-mers you get. Now, if this, whatever this path is compatible with a transcript, it's going to put this read in that transcript. And as you expect, there's going to be many transcripts where it's going to land up in, because 100 is a much smaller number. So that is what it's called, the compatibility class of this read.
And the overlap has to be k-1.
Yeah, the overlap has to be k-1. So if you're taking 30 and 30 here, so the path is determined by that. So yeah, if it is matching the de Bruijn, like if it is contained in the de Bruijn graph of the transcript, then you keep it, otherwise you don't. But here, of course, there's a caveat, you can probably imagine. Let's say there's a sequencing error, like there's many steps that you have done. Like, of course, it's going to be some other path. So what do you do in that case? So what Callisto does is, like, get rid of it. Because that's like a very, that's a very rare phenomenon that's gonna happen, especially if you, oh yeah, by the way, I think I said it takes every single path. So you can imagine if there is a sequencing error or like a SNP or something, it's going to be across the whole gene. So if you have many reads across the whole gene, there's very likely that like many of them will survive. So even if you discard, it's not going to be a big proportion of that. So that's one aspect it uses. And yeah, so that's where you see the K compatibility class of the read is determined by. And using like a binomial model, like you have this, okay, some of my images have vanished, but okay, so effectively, so alpha are the parameters, those are the things we want to quantify. For each transcript, there is a number we want to quantify. And you want to normalize for length, right? Because the longer the transcript is, like you'll get more reads out of that. So you want to account for that. So you divide by this LT, which is this effective length. And by the way, this effective length is actually not the length of the transcript. It's length of the transcript divided by the read length minus 1. Because if you have a transcript like this, and if you have like 100 base read, how many possibilities are there? like how many possibilities of having 100 like base pair read from this 1000 base transcript. There's about, say, like 901. So that's why you use the effective length, because that could make a difference when you have smaller transcripts, because the transcripts range from a length of like in the hundreds to thousands. So when it's huge, it won't make much of a difference. But in that case, yeah, so using like some sort of EM algorithm, you find all these parameters alpha, and that's what is the quantification you have. And so in more recent work, so we performed a comprehensive evaluation of full-length isoform quantification tools. So this is actually a very recent thing, like of having full-length isoform quantification, because gene-level quantification is much easier. Like if I give you a 100 base read, like aligning to the gene level is actually quite easy, and you can be very sure of that. But at transcript level, the issue is, like I was talking about splicing, right? So you can imagine a lot of exons are shared. So if the exons are shared and my read is coming from that exon, I don't know where to put it. So the only real information actually comes from the junctions, so to say. Like when you have two exons, the picture. Yeah, so if you have two exons, if there is a read spanning this, like from here to here, then I'm getting like some information, okay, maybe there is another transcript which actually skipped all this and went here, and the one spanning it, the read is from here to here. So you're able to distinguish. So junctions are the most important part. And until we have full-length isoform sequencing, which is probably in the horizon, like now Pacific Biosystems does it with 85% accuracy, which is still pretty poor, and they're hoping in a few years they'll get up to like 99%. So if we have full-length isoform sequencing, then we don't need all this. Or maybe like on a daily basis, maybe like if you want to use a laptop computer and like do things like this, because even today people use microarrays, although.
What is the longest difference you have observed between two isoforms?
Like highest difference, length difference.
If the exons are chosen,
Mm-hmm.
you would have encountered some isoforms.
Right. Yeah, so I think this will be, because, okay, so since I don't know the answer off the top of my head, I think one can infer this. So let me.
It need not be exact, because,
Yeah, because I'm trying to visualize this, because let's assume that all exons have similar length, and usually they don't, exon length doesn't vary that much. So your question answer, the answer for your question would very much can be answered by saying, okay, what are the number of exons? Like how much do they differ? So number of exons can, the median number of exons I know for all transcripts is like 11, and it can, for a given gene, I think it can go, I have noticed like going from 6 to 15. So you can think like whatever, 600 to 1500, so to say. Yeah, I mean, it can differ quite a bit, the number of exons. And in fact, maybe after the talk you're interested, I have a tool where you can actually look at all these information for a given transcript, like number of exons, length, and all that. So, yeah, so that.
the tool will be able to distinguish a scenario where there are two transcripts versus insufficient raw data. Can identify.
So mostly, the tool I'm talking about is not like pseudo, so it's a different tool. I'll explain it in the end. I'm just saying that to answer your question, that tool can be used. So it is about like getting statistics about all the features of transcripts so that you can make some sort of, like find commonalities between transcripts, like do they all have the same number of exons, do they all have similar length or like GC content and so on. So it's a tool to do that. Yeah, so that's why I said, okay, I mean, I can figure it, like I can find it out. Yeah, anyway, so we did a pretty comprehensive evaluation of full-length isoform quantification tools, which is still pretty rudimentary. And so on idealized RNA-Seq data, like no insertions, deletions, uniform coverage, and all that, Callisto does pretty well. In fact, actually, this is the most popular tool these days. But once you add realistic data, like indels, non-uniform coverage, and I'll mention what this non-uniform coverage means. So accuracy for genes with more than 3 isoforms is very significantly reduced, to the point that it's useless. So I don't know if a biologist would ever be interested in, all genes which have just 3 isoforms. I don't know. So in a way, one message would be, okay, if you want to do gene level analysis very quickly, maybe you can do Callisto and merge all those to get the gene level expression and do your analysis. It's going to be quick. And in fact, Leopactor has a tool where it says, write a paper in 4 minutes. And how do you do that? Go to Geo. Geo is like a gene expression omnibus where you have a lot of data. So you can run Callisto in 4 or 5 minutes for n number of papers, and you can get the kind of list of genes which are differential and do your standard analysis and do that. Of course, he was half joking when he said that. But the point being is that if you get a fresh data and you don't want to wait 2 days, maybe you run this and get a rough idea of what's going on, then just move on, or maybe investigate further. But for full-length isoform quantification, we are not so sure. And just to show you what idealized coverage and realistic coverage, what's the distinction? So idealized coverage would look like this. Effectively, it should be flat, but if you're random sampling, then kind of wavy pattern. And the ends, because there's only so much you can have at the end. So that's why you have this drop-off. But when you do realistic coverage, there is a lot of biases. It doesn't look anything like that. And there are many speculations about why this happens. And one of the works we have been doing, I've been talking about a few things, and I'll come back to alignment at the end. So this is one of the things our lab has been doing, so I've been involved in that. It's an RNA sequencing simulator, and that is very important to create good benchmarking data, because a lot of tools are being created, but they're always tested on very simplistic data. For example, maybe we don't know the biological mechanism which is causing this. We have some ideas, but we don't exactly know. But we have to kind of capture this information when we create our simulated data with the known truth, so to say. So we talked with some Illumina guys, and they suggested, okay, maybe there's a lot of RNA secondary structure that's going on. So when you're doing a fragmentation, there's a lot of fragmentation bias. But we tried to, and RNA secondary structure is actually a very hard problem. There are many tools out there which, if you give a sequence, it will try to predict the RNA secondary structure, somewhat optimizing for free energy and so on. But that does not seem to be any correlation. We use those tools and try to correlate with the, let's say, the heights, so to say. So it's not very clear. But yeah, we're doing more experiments about that. And just to show you how complicated the picture is, usually you start with full transcriptome, and then you select, you want to remove your ribosomal RNAs. You don't want them, because in RNA sequencing, you want the transcripts. So you do a poly selection, and that introduces some bias. And because RNA is not that stable, that's why it's useful. So DNA is more stable. So first you convert to RNA to DNA, then you do all these steps, PCR amplify, and then eventually you get the list of reads I was talking about, 100 base pair reads and so on. So final step is list of reads, but you can see how many steps have biases in them. So ideally, we would like to know all of them, but of course, that's probably, each bias probably would be one research group or something like that. So we are creating a modular framework where if you get a new model, you can just move that module out and move your module in. So that's called BEERS. So benchmarking for effectiveness of RNA-Seq software. So that's what we have been working on. Okay, so I think I'm running out of time here. So I'll just quickly talk about multiple sequence alignment. So we talked a little bit about pairwise sequence alignment. So with multiple sequence alignment, you can use the same, you can think about the same problem. Instead of having a matrix kind of structure, now you have a 3D structure. And you do the same dynamic programming kind of algorithm. You have this pair, and first you fill this up the plane, and then you keep going, and there you had, the recursion had 3 things, right? You had 3 ways to come there, and you chose the maximum of that, but here you'll have 7 ways. because it's like a cubic kind of thing. So the number, you can see that like if I'm trying to align multiple sequences, the problem is just gonna get out of hand. So what people use is other kinds of techniques, like for example, a hidden Markov model. And so just to remind you what a hidden Markov model is, so you have a bunch of states evolving as a Markov chain. So you know, sometimes you know what the transition probability matrix is, but what you are not able to do is actually observe the states. And so there are some other related things you can observe. And you know the initial distribution of states. And so there are two problems here. Like first, let's say all this information is given to you, and the transition probability matrix, the observation matrix, so to say, and the initial distribution of the states. Then you can, like looking at the observation sequence, now you have a sequence of observations. So that probably corresponds to a sequence of states. So you want to predict like, okay, what's the most likely sequence of states that resulted in these observations? And then there's another kind of problem. Let's say you actually want to construct this kind of transition probability matrix which explains this kind of observation. So there are two problems, and both of them are actually important in multiple sequence alignment, because this will be used to train your model, and this will be used to actually use the model. Problem 2 will be to train and actually get the hidden Markov model, and problem 1 to actually, whenever you get new information, you want to use this model to like gather whether it's aligns or not. So what is used in like multiple sequence alignment of proteins to get protein families, so to say, like protein family domains, is effectively a Markov model which looks like this. These are called matching states, these are insertion states, and these are deletion states. And these numbers are kind of ad hoc, but like people play around and figure out like, because you have to specify the number of states beforehand, before you can like build the hidden Markov model. You need to know how many states are in there. And for each of, this is a start state, end state, and for each of them you have just one delete state, because of course if you did like 4 deletes and you have 4 things in the sequence, it's just all gone. And just to give you an idea, so here, so if each state emits one specific amino acid with probability 1, that means you observe one specific amino acid, emits means that, and the transitions between the match states is 1. I mean, so insertions do not happen exactly like you match everything, so the model always produces the same sequence. So in that like model, there's only one sequence. And if you remember, I was talking about like multiple sequence alignment. There's this abstract like way of aligning. You're not really aligning to anything, any particular sequence, but rather here you're aligning to a model. Like you have a model for this family and you want to see whether you belong to that model or not. So, and of course there's and the other example was if match and insertion states are uniform over the 20 amino acids, then model produces any sequence you want. So what you do is actually empirically figure out like all these protein sequences and then build your profile HMMs. And then if you get a new sequence and you want to figure out what like, which family does it lie in, which family of proteins does it lie in, you run it through this HMM. And then the results you get are something like this. Suppose like you start with a 10 match states, let's say you have these things. And let's say, like when you applied the profile HMM on this, you got M0, M1, M2, M3, blah, 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 and all the way. No insertions, no deletions. So this. But let's say when you ran this through the hidden Markov model, you got all of these. So then you have a deletion here. So like whatever the path, the most likely path it has traversed in that hidden Markov model is, will give you this alignment. Like for example, here you see 2 deletions, which is why you have 2 things here, and so on. So this is what like a multiple sequence alignment looks like. Actually, yeah. So one of the concrete problems, like, so I mentioned I worked at ITMAT, which is the Institute for Translational Medicine. So one of the things we really like to work on is, like usually when you do an experiment, you end up with a list of genes, like especially in high throughput like sequencing studies, you end up with a list of genes or transcripts. And a lot of the times, then you start validating using PCR, or like maybe you have a biological hypothesis and so on. So we want to help like people in like forming all these biological hypotheses. So looking at those set of genes, we want to see what's common amongst them. So one thing to do is we need to have a database. Like for all these genes, we like have number of exons, lengths. Are those the common things? That's coming back to your question. And it could be entropy, it could be compression complexity. Like compression complexity would just mean like if it's like a trivial sequence, it will be highly compressed. Like you can compress it very much. So like things of this nature. So we have a database which does this. Like it has information for each species, and looking at all genes, it has all this information. And then when we have a list of genes, then we see if there is some enrichment there. And so that like we can form some sort of hypothesis. And as I mentioned, correlation does not imply causation, but it's a start. It will give you some hypothesis. And yeah, so going back to that Callisto study, so one of the things we noticed is that sequences which Callisto does really poorly at, you can see that for the population mean, for example, is around here, like the black line. So this is the transcript sequence compression complexity. And the things where Callisto did, we isolated all the transcripts where Callisto did badly. And as you can see, the median line has kind of shifted here. So effectively, it's saying something which is intuitively obvious, that things which were less complex, Callisto was bad at actually identifying, which you would expect. Like if there is less complexity, all this information that has been lost, it will map to multiple things. If it won't map to something uniquely. So this is one way we figured, okay, I mean, there's something going on there. And the other thing is, let's say exon count. Like exon count, like the median is about 4. Like for taken across all genes. And here, for Callisto, it's actually, the transcripts where it's doing badly is actually around 9. So which means that like the longer the transcript is, Callisto is doing worse, which you would also expect because, again, like there's the uniqueness issue, how to map things. Okay, and the next steps we have in mind is actually like taking like pathways and yeah, I mean, like we have the database ready, like just to, in fact, like we have a website for that, like it's called tequila.org. So the other thing is we want to actually like take genes in different pathways, let's say 20, 30 genes like cardiovascular inflammation, this or that, and we want to create like HMMs for that and see like how it relates to other things, like do some sort of sequence alignment of at the gene level and like whatever like information we get, like we want to like form hypotheses based on that. Okay, I think this is, okay, thank you. Mm-hmm.
Of the many possibilities, only few transcripts are realized.
Right, yeah.
So one could argue that other transcripts are yet to be realized. They are not there.
Mm-hmm.
Maybe our collection of the RNA did not pick it up.
Mm-hmm.
But other is there is an inherent risk combined to form a transcript.
Mm-hmm.
What could decide that?
Actually, yeah, I wouldn't know the answer, but like, yeah. Yes. So no, I was going to add something to that. Like, yeah, it is observed that even like the maximum number of exons I have observed is like 44. I think there is a gene which has 44 isoforms. So, but that's a very rare case. You like the median value, as I was saying, is like 11, and even out of so sorry, I mixed both. That was the exon count. So 44 was number of isoforms. But even then, like the actual number of isoforms that are expressed is like 4 or 5 max. Like very rarely, like unless you actually do a knockout experiment, okay, I knock out this exon. Unless you do some experiment like that, you'll never really see more than 4 or 5 isoforms really being expressed. And whenever, the other issue is whenever we see this 20 isoforms or something, that's mostly because of annotation issues. Like, because the annotation we use is from the team called Ensemble, and they're not at all conservative. Whatever, for example, they will see that there are 200,000 transcripts and 50,000 genes. Like whatever they get from the biology, they don't really like empirically investigate. Whatever they get from the alignments, they create an annotation based on that. So they are the exact opposite of conservative. So, so your question was about the mechanism. So first, I'm prefacing that with saying that, okay, it is true that very few are actually ever expressed. And mechanistically, of course, like the first and last exon, of course, you expect, like the first exon you expect to be there because the Kozak sequences, which, like you need a Kozak sequence to initiate the translation, right? So, and it has to be, I think, about 10, 15 base pairs away from the start codon. So you want that. So it cannot be arbitrary. Like that cannot be removed, for example. So I would imagine like some of those exons are probably have to be there, but otherwise, yeah, I don't know really. Do you have like a...
Well, I don't know. One possibility is also mentioned. More stable, so
like terms of secondary structure, you mean? Yeah, it could be because when splicing happens, like it looks to me a very stressful event, right? Like you have this massive intron, like a small exon, although the diagram didn't show to scale. Then you have this massive intron. And before the splicing happens, it warps around, and of course there are signals here, and so on. So, yeah, I mean, the secondary structure will have a role, I would imagine, in that sense. So,
theory part tomorrow?
no, actually tomorrow will be a completely different talk, but yeah, I mean, we can talk about that like if you want later. Because yeah, I realized like that's something that needs a talk of its own, like because just setting up notation would like, yeah, take a lot of time. Like categorical aspects, like, so more recently, this is actually this year itself. So Remy Tueras, he's a postdoc at MIT. So he has been writing a series of papers called Category Theory for Genetics, like where he is actually, so multiple sequence alignments and all, and not just like insertion, deletion, this, but inversions and like recombinations. So he's developing a logical language for that, like in terms of Kahn extensions, like adjoint functors and so on. So that is also something quite interesting. I'm trying to learn it, like I'm still a student. I don't think I will speak from any point of authority, but I can let you know, like what he's doing. Yeah.
There's someone,
David's
Grist or something like that.
Bob Grist. Yeah. He's at UPenn actually.
UPenn.
Yeah. But he does more like what's that called? Like sensor networks, applied homology theory. Like
Right.
Yeah.
But that also, so I remember it was one of my classmates in grad school was Sanjeevi Krishnan. Worked with him on some data grants or something.
Yeah.
But applied side of category theory, I'm not sure if it was.
Yeah, it was more like you have all these like nerves of coverings, like you have all the sensor networks. You want to like do some.
In the biological sense, not
No. I mean in the
yeah,
in the mathematical sense. Yeah. So you have this graph based on the like the range of your like sensors, and you want to ensure there are no holes. So like
this is more in the engineering.
Yeah, more in the sensor networks, like your hole detection effectively, and yeah.
So again,